Alright, so today we're going to go a little bit further. We're going to go into another process that the cell does using that information we talked about yesterday, the DNA, RNA. Alright, so looking at that, to understand that first though, we need to know what a gene is. Alright, so it says here a gene is a segment of DNA that codes for a certain protein. So we talked about proteins already. And proteins are what your body uses as pretty much instructions for doing things. Okay. We know that ribosomes make proteins. All right, so we're going to have the ribosome involved here to make that protein from that gene. Okay. The process of doing that is called transcription. And that's what we're going to look at today. So we're going to take that DNA sequence, that chunk of DNA that we had, we're going to turn that into mRNA, messenger RNA, and then through the process of transcription, turn that into a protein eventually in the ribosome, okay, after transcription. So transcription itself, we have 5 basic steps for transcription. First off, just like replication, that DNA helicase comes in and it unzips the DNA. So instead of having 2 connected strands here, we're going to take that helicase and split those bonds, those hydrogen bonds, and open up that DNA again so that we have 2 single strand sides. Okay. Instead of pairing that with free nucleotides for DNA, we're going to pair that with mRNA nucleotides. Okay. So RNA is a little bit different. We got some differences, remember. We got T's, U's instead of A's, things like that. Now, what builds this is RNA polymerase. Right? Got to remember that. DNA polymerase builds the new DNA strand. RNA polymerase builds RNA. Okay. Once that happens, and we get that single strand, that double helix goes back together, stays inside the nucleus. All right. What we're finished and what we get when we're done is a strand of mRNA. It's only coded for certain genes that need to be coded for. Okay. Now, why do we have to do this? Why does the cell have to make this messenger RNA? It's real simple. Your DNA is in the nucleus of your cells and it never leaves. Does not leave the nucleus. DNA is important. It is the most important piece inside the cell. It's kind of like if you have $1 million. You don't just leave it laying on the porch for anybody to walk by and steal, or something happen to it, get wet, burn up if a fire starts. You put it in a safe. Well, the same thing with DNA. It's that important. It's put into that nucleus protects it, and it never comes out. So in order for us to make protein in the ribosomes, that DNA has to come out. Well, the only way it can come out is if we make a copy of it. Well, the copy we make is called messenger RNA. And that comes out of the nucleus so that we can we can make proteins. Okay. So here's what the process looks like. We have our double helix. It opens up, just opened up for that gene. That's it. The rest of it stays coiled up. Stays nice and compact. You have your helicase that split it open. You've got your RNA polymerase that comes in and makes that single-stranded mRNA. All right. Similar to DNA, single-strand DNA, except we got the U's in here, and it doesn't make another copy and stick it together. It's just one single strand. All right. So this is kind of similar to another process we've already talked about, DNA replication. Okay. So that replication process, just leave my face there, is similar. Okay, they both involve the 2 enzymes. For replication, we have DNA helicase. Here we have our helicase. We use the same helicase for transcription as well. Okay, we use a polymerase, except for replication, we use DNA polymerase. Transcription, we use RNA polymerase. So we have those 2 two enzymes that come in and do this work. All right. The difference is what we get out of it. For replication, we get a copy of DNA. For transcription, we get a copy of just one single gene. Okay. So if you look over here, this is a microscopic view. All this stuff is DNA. These are all chromosomes. All right. But if you notice, we have this one right here in the center, and we have a piece that's coming off of it. That piece that's coming off of it is our mRNA. That's where it's making that mRNA. It looks similar to DNA, but that chunk's going to be able to leave the nucleus, whereas the DNA has to stay in the nucleus. And you can also see it down here. This is uncoiled. All right, so all of this is uncoiled. And if you look right down here, you see these single little lines. That's when that DNA is twisted up and shrunk and compact. Big difference. One little single line that's very tiny compared to that big leaf, tree, bush looking thing when the DNA uncoils. Okay. Alright, so we have our bases. We've got our A's, G's, C's. Ooh, no T's. There's only U's there. Alright, so there's no T in mRNA. Remember that. And we look at each 3-base sequence. So every 3 bases is called a codon. So we have 1 base, 2 base, 3 base. That's called a codon. All right. Each of these codons codes for a certain amino acid. Amino acid are the building blocks of a protein. Okay. So we get one amino acid here. This one could code for methionine. All right. It could code for lysine, glycine, glutamate. I mean, there's a bunch of different ones, and when you go back into Canvas and you look, there's going to be a chart that you need to download, and so that you can translate tomorrow this mRNA into a protein. All right. So each of those 3 bases, each codon codes for one protein. We could have several different codons that code for the same protein. It's just a certain order of those bases tells us what amino acid is going to be put into that protein. Okay. When we take all those amino acids, so we have 1 codon, 2 codons, 3, 4, 5, all the way down. Each of these codes for a certain amino acid. So we're going to have all these amino acids put together. When we get the whole thing put together, we have a protein. So all of this put together would make a protein. That process of making the protein is called translation, and that's what we're going to go over tomorrow. All right. So transcription, it's pretty simple. It's similar to replication. Uses those enzymes as well as replication, but now we're making RNA. No T's, we got U's instead of T's. Okay.